Well, we have the opportunity to present the results of about 5 years worth of work. It is called ABLE Studies, which is Applied Biomarkers and Late Effects. It's a pediatric study funded by the Canadian Institutes of Health Research, the CIHR, through the consortium in the United States and Canada, which is called the Pediatric BMT Consortium. What we did was we did a prospective study of over 300 children and have now done an analysis and identified both looking at biomarkers to help us risk stratify children, but what we'll be talking about at the meeting here is about the biology that we learned about. And the biology we learned about was that we've always known clinically there's different types of graft versus host disease when you look at the late time points. There's what we call late acute GvHD, that the NIH consensus group has called it that, and the second is chronic graft versus host disease, which we all know about. And we were able to do an analysis of the biomarkers and do a very comprehensive analysis of phenotyping, cell populations, looking at plasma markers that are known in graft versus host disease, look at clinical factors, and we found that there were patterns. And what we have shown now is that late acute graft versus host disease, which we've always known is a different disease than chronic, actually has a biological pattern that's different as well. And we think that some of the information that's going to come out of this is not only going to allow us to do biomarkers to guide therapies, the current treatments we have, but also to identify targets for treatments in the future. And one of the big findings that I'll present at the EBMT meeting is that of that the loss of regulatory populations is characteristic of chronic graft versus host disease where it's not in late acute.